students, in this lecture we are going to discuss the pollen-pistil interaction seen in angiosperms. In our previous lectures we have already studied that the different mechanisms of the pollination, different agencies of the pollination, and how exactly they are working. In this lecture we are going to discuss how exactly the pollen grain is interacting with the carpel. In case of angiosperm, the pollination is considered as indirect pollination, and it is the important characteristic feature of all angiosperm. Now let us discuss why this pollination is called as indirect pollination. In case of angiosperm, when pollen grain travel to a long distance to reach up to the carpel, it will first received by the stigma. So the pollen resting site on the carpel is stigma, and therefore the compatible structure to the pollen is not ovule in case of angiosperm, it is the stigma. So the stigma is serving as the compatible structure for pollen grain. Now you can observe in the diagram, pollen grain in angiosperm is not coming in direct contact with the ovule. Ovule is deeply situated in the ovary and pollen is resting on the stigma. As there is a much gap, they are not coming in contact with each other. The pollination is considered as indirect pollination. Then, what do you mean by pollen-pistil interaction? So pollen-pistil interaction, in short, it is nothing but these are the serial events occurring right from the deposition of pollen grain on the stigma up to the entry of pollen tube into the ovule. All these events which are occurring in between these two is ideally called as pollen-pistil interaction. Let us discuss one by one. When pollen grain is received on the stigma of the carpel, the first thing which is done is pollen recognition is done by means of carpel. Now what is the pollen recognition? Stigma or the pistil, or what we call it as carpel, the female reproductive structure, first recognizes the pollen. The pollen of having same genotype, as we have discussed in self-sterility, which is the genetic mechanism. When genotype of the carpel and the pollen is same, such pollen grain is rejected by the carpel. It will not allow to germinate on the stigma. So in this way, the first event in the pollen-pistil interaction is recognition of right type of pollen, and that is done by means of stigma. When wrong type of pollen, it means pollen from other species. Say for example, this is the pollen from other species. This carpel is belonging to the X species and this pollen belongs to Y species. Now this pollen is from unwanted source, not a regular type of source, belonging to different species. Now such pollens are not allowed to germinate. Now the pollen which is selected by the stigma is from the correct source, or correct type of pollen is selected. How it is that? Let us discuss. Suppose these two pollen grains shown in red color, they belong to the same species and the species X. But here, suppose the genotype is S1, and here in this case the genotype is S1 and this is having S2. So the right type of pollen, most favorable type of pollen in case of germination is S2. So from unwanted source, pollens are not allowed to germinate. The same kind of pollens belonging to same species, they will germinate, but the growth of their pollen tube is stopped. It is prevented, and the correct type of pollen is selected for germination. It is successfully germinated on the stigma and able to produce the pollen tube. The growth of pollen tube is continued, and the pollen tube will run from stigma to the style, finally enters into the ovary and is capable to enter into the ovule. Now, as we have discussed, the pollen tube is attracted towards the ovule by means of a structure present in an embryo sac, and that structure is called as synergids. These synergids are having a special type of structure, what we call it as filiform apparatus. This structure is attracting to the pollen tube towards the ovule and is able to enter inside the ovule. Now, the movement of pollen tube along the style, through the style towards the ovary is called as chemotrophic movement. This part is very important. The movement of pollen tube is considered as chemotrophic movement. The reason behind that is the pollen tubes move along the pistil or move along the carpel along the gradient of calcium, boron and inositol sugar. This is very important. The attracting structure or this pollen tube is running towards the concentration of calcium, boron, inositol sugar. So this part is very important. You have to remember this because this can be asked for your competitive exams like NEET. So calcium and boron, these are the elements are very important for a pollen to germinate and to form the pollen tube. And along the gradient, the pollen tube is running through the stigma towards the ovary and ovule. So calcium, boron, inositol sugar concentration is very important. Pollen tube is running towards the concentration gradient and therefore the movement of pollen tube along the pistil or from the pistil is considered as chemotrophic movement. On the other hand, the pollen tube at the time of running through the style and growing towards the ovule is responsible for secretion of certain phytohormones, and those phytohormones which are secreted by the pollen tube are auxins and gibberellic acids or gibberellins. Now, these phytohormones are the growth promoting structure. They are well known for cell division. Now see, this part is very essential to understand. The pollination is considered as the signal for fruit formation. You are already aware of that after pollination, fertilization occurs, and after fertilization, the ovary is converted to fruit and ovule is converted to seed. At the time of fruit formation, the small ovary is increased in the size to produce the fruit. And that signal of fruit formation or enlargement of the ovary is mainly given by auxins and gibberellins secreted by the pollen tube. These are the growth promoting substance, as we have discussed earlier, are responsible for cell division in the ovary and thereby increase the size of the ovary and convert it into the fruit. And therefore, the signal for fruit formation is pollination because pollen tube is responsible for secretion of auxins and gibberellins. Due to secretions of auxins and gibberellin by pollen tube, ovary is stimulated to secrete the same phytohormones and it will increase in the size, and thus ovary start growing into the size which is called as fruit formation. And therefore, in practical application in horticulture as well as in agriculture, you can replace the pollination by auxins and gibberellins. One can spray auxins or gibberellin to produce the fruits without fertilization, and what we call it as parthenocarpic fruits. It means when we spray auxins or gibberellin on the carpel, on the flowers before pollination, you can achieve the fruit formation without pollination and fertilization. On the other hand, one very important factor you have to study is in parthenocarpic fruit, the fruit is developed without fertilization and therefore ovary will increase in size to produce the fruit, but ovules are not converted to form the seeds because fertilization is absent, and therefore parthenocarpic fruits are seedless fruits. Important points I have to revise here. Listen carefully. In pollen-pistil interaction, the important mechanism you must know is indirect pollination because pollen grain and the ovules, they are not in direct contact with each other. Second important thing is the serial events occurring between deposition of pollen grain up to the entry of pollen tube into the ovule. This is called as pollen-pistil interaction. Stigma selects the right type of pollen. It means the same genotypic formulation of the pollen is rejected and not able to produce the pollen tube. Secondly, pollen from other sources or different species, they are not selected by the stigma and they are rejected. pollen from ideal source and with a different kind of genotype with respect to gene S is selected and is able to develop the pollen tube, and the pollen tube will grow along from the stigma to the style towards the ovary, and this movement is called as chemotrophic movement because the pollen tube is growing along the gradient of calcium, boron and inositol sugar. In this way, the pollen tube will subsequently enter into the ovary and finally enter into the ovule through micropyle. Next important thing is, pollen tube at the time of growth is responsible for secretions of phytohormones like auxins and gibberellins. Now, auxin and gibberellin will stimulate the secretion of the same phytohormone by the ovary, and in this way the cell division start in the ovary will increase in the size and fruit is formed. Formation of fruit, the signal of formation of fruit is given by pollination, hence we used to call, and fruit formation is achieved after pollination. You can replace the signal of pollination by applying auxins and gibberellins because these phytohormones induces the increase in the size of ovary without fertilization and without pollination, and in this way we can develop the seedless fruits, what we call it as parthenocarpate fruits. Now let us discuss the course of pollen tube entering into the ovule. We have already discussed that pollen tube is produced in the stigma region. It will run through the style, finally enter into the ovule, most probably through micropyle. But there are different chances, different patterns of entry of pollen tube into the ovule. Let us discuss one by one. Suppose this is the right type of pollen selected by the stigma for germination. It will produce the pollen tube, and this pollen tube will run from stigma then to the style, finally enter into the ovary and then enter into the ovule through micropyle, which is the very common course of entry. Now this is denoted here, the first type. You can say entry of pollen tube is generally from micropyle, and this type of course, this type of mechanism is called as porogamy. The most common way of entry of pollen tube into the ovule is porogamy, but sometimes the path may differ, the path may vary. Sometimes, in some cases, this pollen tube directly enter through the chalaza inside the ovule. Now this is the second one. When pollen tube enter through chalaza inside the ovule, this mechanism is called as chalazogamy. The third type, the third pattern, third variation of entry of pollen tube, sometimes the pollen tube will run like this and it will enter into the ovule through integuments. So which is very different than that of the porogamy as well as chalazogamy. So this path is the third path of the entry of pollen tube, and in this type integuments serves as the entry point for the pollen tube, and this is called as mesogamy. To remember, it is very easy because micropyle is the minute pore. When pollen tube enter through micropyle, it is called as porogamy. Easy to understand, easy to remember. Chalazogamy, the name itself is having the term related with the chalaza, and therefore chalazogamy, it is also easy to remember, easy to recollect the concept, but you have to focus on mesogamy because integument such terms are not mentioned in the phenomenon. Mesogamy, meso in between. Now the entry point is through integuments, which is in between the micropyle and chalaza. So you can compare it with when pollen tube is entering through integuments, at that time it is called as mesogamy. In short, pollen tube can enter into the ovule by three different methods. Most common is porogamy. Chalazogamy is entry of pollen tube through chalaza. Mesogamy is entry of pollen tube through integuments, but these two are occasional, very rare, and most common type is seen, which is called as porogamy. One more important concept here we are going to study is when pollen germinates, it will form a pollen tube. We have already discussed the development of male gametophyte. Now this pollen tube consists of a tube nucleus, cytoplasm, vacuolated cytoplasm, and it also shows presence of two haploid male gametes. So these are the male gametes. The male gametes, in case of angiosperm, they are traveling through a tube. The tube is called as pollen tube. Tube in Latin is called as siphon, and therefore traveling of male gamete through pollen tube or in pollen tube is considered as siphonogamy, and thus siphonogamy is again the important characteristic feature of angiosperms. By the time we have studied the different steps involved in sexual reproduction in angiosperms, such as microsporogenesis, megasporogenesis, development of male and female gametophyte, pollination, and now this is the time to study the fertilization. In angiosperm, the characteristic type of fertilization which is seen is double fertilization. Before discussing what is double fertilization, you must know what is fertilization. Fertilization is nothing but fusion of male gamete and female gamete. To produce the diploid zygote. The ploidy of gamete is haploid, and formation of the zygote, which is diploid. So fertilization, the definition is very easy. The process of fusion of male gamete and female gamete to produce diploid zygote is called as fertilization. But in case of angiosperm, fertilization is seen in two different steps. Two different fertilizations are seen, and therefore the process is called as double fertilization. In case of angiosperm, in the first step, the male gamete fuses with female gamete to produce the zygote. This is the first step involved in the process of fertilization. And the second step, in case of angiosperm, where another male gamete, it fuses with the secondary nucleus. A male gamete is having a ploidy, that is haploid. Secondary nucleus, you have already studied that it is diploid. The fusion, the product of fusion is triploid, and it is called as primary endosperm nucleus, abbreviated as PEN, P-E-N. It is triploid. So such two steps are seen in case of angiosperm during their fertilization, and therefore the fertilization is called as double fertilization. So let us see the definition. Double fertilization is a process in which one male gamete fuses with female gamete or egg to produce the zygote. Other male gamete fuses with diploid secondary nucleus to produce primary endosperm nucleus. This is called as double fertilization. Double fertilization in angiosperm was studied by Navashin in 1898, especially in the plants like Lilium and Fritillaria. So these are the well-known plants in which the double fertilization process is first time seen, and it was discovered by Navashin. The process of double fertilization in angiosperm is studied in two different steps, in two different mechanisms, and those are syngamy, the first mechanism in double fertilization. Here the term syn, it means fusion. Gamy is related with gametes. So it is very clear that when pollen tube enter into the ovule, as we have discussed the more common path of pollen tube, it is running like this, entering into the ovule most commonly through the micropyle. So this pollen tube is carrying the gametes. Suppose this is the pollen tube, it is entering inside the ovule, and the attracting structure to the pollen tube is filiform apparatus, which is associated with synergid, and therefore pollen tube will come in contact with one of the synergid like this. The entry of pollen tube into the synergid is responsible for degeneration of the synergid, and after degeneration of synergid, the content, cellular content, cytoplasmic content of synergid is absorbed by the pollen tube. It will swell in size and burst thereby to release the male gametes. By the time the tube nucleus which is present here will degenerate, this pollen tube will burst open, synergid will degenerate, and what will remain here after degeneration of tube nucleus and synergid is nothing but two haploid male gametes, and those are released. This is first male gamete and this is the second male gamete. Two male gametes are released here. Now, one of the two male gametes that will come in contact with female gamete, which is nothing but egg, and fusion of male gamete with egg is responsible for formation of zygote. Male gamete and female gamete, that is egg, both are the gametes and they are showing fusion, and therefore the first event, first step in double fertilization is called as syngamy, where the male gamete fuses with egg to produce the zygote. So this is the first event. The second event, where, as we have discussed, the second male gamete will come in contact with the diploid secondary nucleus which is present at the center of embryo sac. So this is another male gamete is coming in contact with the secondary nucleus. So it is like this: male gamete plus secondary nucleus is responsible for formation of primary endosperm nucleus. Here male gamete is haploid, secondary nucleus is diploid because it consists of two haploid polar nuclei, and therefore this is nothing but fusion of three haploid nuclei, and hence this event is called as triple fusion. Let us discuss the significance of double fertilization. First and foremost significance is fertilization or double fertilization in angiosperm is responsible for formation of fruit and seeds. Second important thing is by means of syngamy, where male and female gamete fuses with each other and form the zygote. This zygote divide and redivide to produce the embryo which will develop the further generation. The third important significance is in case of double fertilization, the event triple fusion is responsible for formation of primary endosperm nucleus. This nucleus or this cell will divide and redivide to produce the nutritive tissue, what we call it as endosperm. This endosperm provides nourishment to the developing embryo present inside the seed. Next significance is a restoration of normal diploid number in the progeny. Why this is so? Because in case of syngamy, haploid male gamete fuses with haploid female gamete or egg. Both the haploid structures, they fuse together to develop the diploid zygote, and therefore the normal diploid number of the species is continued in the rest, is continued into the generation. Very important characteristic feature or significance of double fertilization is here recombination is seen, because fusion of both haploid structures, basically these haploid structure or gametes, they are produced by meiosis, and you are already aware of that. Meiosis is showing recombination, genetic recombination. Now these structures are again fuses with each other, so genetic material of both the gametes will mix with each other, what we call it as recombination. This recombination is important to develop the variations in the progeny, and variations are very strongly supportive to the process of evolution, and so double fertilization is evolutionary important. Last significance of the process is this process utilizes both the male gametes entering into the embryo sac. One is fusing with egg, another is fusing with secondary nucleus, and thus this process is important to prevent the polyembryony. Single seed used to produce single embryo. There are two different male gametes, but only one embryo is formed. The reason behind that is utilization of both the male gametes in the process of double fertilization. Here one point is very important. In case of some angiosperm, more than one embryo is also seen, and that is called as polyembryony, and there are various different reasons for that. We are going to discuss this point in the next lecture. But importantly, double fertilization is responsible for preventing the polyembryony by utilizing both the male gametes.